students, today I will introduce you with the subject biopotentials, bioelectricity or electrophysiology. It refers to the electrical phenomena of life processes which controls all the organs of our body. So as to apply AI and machine learning in biological system, it is very essential to understand we should have knowledge of bioelectric phenomena. So let us come to first slide. Now, biopotential, generation and propagation. Biopotential are the potentials generated inside the body due to electrochemical action of the cells. They serve to control the various functions of our body. Brain acts as central computer, receives the signal from various parts and organs of our body in the form of bioelectric signals and sends the command again in the form of bioelectric signals. Various types of bioelectric signals are generated due to electrochemical action of the cells. Now, let us come to the point, what is the source of these signals? We know that our body is made up of cells. The living substance inside the cell is known as protoplasm. Approximately 75% of the protoplasm is water. The protoplasm is actually a complex colloidal solution having several organic and inorganic salts dissolved in it. And we know that in dilute salt solutions, the salts dissociate into ions. As for example, sodium chloride will dissociate into sodium plus and Cl minus. Similarly, KCl will dissociate into K plus and Cl minus. Let us discuss what is actually the source of the signals. Basically, these ions are responsible for carrying charges in our body, that is any tissue. And we know that internal environment of a tissue has cells and extracellular fluid surrounding it. All the tissues are made up of cells and the extracellular fluid around it. So, the cells are having a semi-permeable membrane which is insulator in nature. Now, a difference of potential exists between inside of the cell and extracellular fluid outside the cell at resting state. Or resting means when the cell is not excited or it is not working. It is known as resting potential, and the value of resting potential is minus 75 millivolt for a nerve cell. So, the difference of potential exists between the inside of the cell and the extracellular fluid surrounding it. Now, so let us come to resting potential. A potential difference usually exists between the inside of the cell and the extracellular fluid surrounding it at resting state or unexcited state, which is called the resting potential. Now, the function of the cell can be controlled by the magnitude of this potential. The value of the resting potential is fixed for a particular type of cell for a particular species. So, it is the specialized role of nervous system to propagate the membrane potential within itself and to transmit them to other cells. So, we can record this. This is inside of the cell and this is the extracellular fluid, and by inserting one of the electrode inside, and one in the extracellular fluid, we can record the difference of potential between the two compartments. And we know that we are doing recording of the neurons. Neurons are the nerve cells having several processes. Some small processes like dendrons and a big process called the axon. So we are keeping the electrode here, and for the in vitro experiment we are keeping the whole neuron in a sodium chloride solution and keeping one of the electrode, inserting one of the electrode inside and another outside the cell and recording the difference of potential. And it is approximately, it comes to minus 75 millivolt for the nerve cell. So at resting state, there is always a difference of potential between the inside of the cell and extracellular fluid, and this is around minus 75 millivolt. And it is the specialized role of nervous system to propagate the membrane potential within itself and to transmit them to other cells. So what happens whenever the cell is stimulated, there is change in the difference of the potential, and this is then propagated throughout the cell and to the neighboring cell. And the changes in the value of resting potential is regarded as the unit of information in biological system, and that helps to coordinate the action of various group of cells or various organs. So resting potential is minus 75 millivolts for the nerves and minus 60 millivolts for the muscles. Now, let us come to origin of the resting potential. So, we have come to know that there is a fixed resting potential for a particular type of cell for a particular species, and if any change in the value of the resting potential take place, which can only happen when the cell is stimulated, that acts as unit of information because it is the specialized role of nervous system to propagate the any change in potential or any change in the value of the resting potential within itself and to the neighboring cell. So now let us come to the point, the origin of the resting potential, how this is originated. The charge disequilibrium, which is expressed by resting potential, indicates certain excess of anions or negative charges inside the cell and corresponding excess of cations outside the cell. The cell membrane can be regarded as the electric capacitor in which two conducting media, intracellular fluid and the extracellular salt solution, are separated from each other by a non-conducting layer, the cell membrane. So suppose this is the inside of the cell and this is the diagrammatic presentation of both the chambers. This is inside of the cell and this is extracellular fluid, and they are separated. Both are good conductors and separated by the insulating cell membrane. So the insulating cell membrane is 6 nanometer thick. In order to charge a capacitor with this plate spacing to the resting potential of minus 75, approximately minus 75 millivolt, approximately 500,000 ions are displaced per cubic micrometer of the cell. So, if we see or if we check the distribution of ions in 1 micrometer into 1 micrometer into 1 micrometer area, 1 cubic micrometer area, we find that potassium, this chamber codes for the inside of the cell and this is the extracellular fluid. So, concentration of potassium ions in inside of the cell is 100,000. In extracellular fluid, it is 2,000 only. Sodium ions are 10,000 in the intracellular fluid and in the extracellular fluid, there are 108,000. And in inside of the cell, and 107,800 anions are provided by the proteins which are present in dissociated form, and they are balanced by the chloride ions 2200, and this whole anions are balanced in the extracellular fluid by 110 thousands of chloride ions, which can freely move in both the direction. So we see that inside of the cell is rich in potassium ion. Its value is 100,000 inside. Now, distribution of ions. So, the source of resting potential is the unequal distribution of the ions inside and outside the cell, particularly the potassium ions. The intracellular potassium concentration is 20 to 100 times greater than the extracellular fluid. Similarly, the intracellular sodium concentration is 5 to 15 times lower than the extracellular concentration. And the distribution of chloride ion is reciprocal to that of potassium ions. It can move freely in both the directions. Now, so this is the diagrammatic presentation. This is inside of the cell. This is the extracellular. Potassium is more in the intracellular fluid and less in the extracellular fluid. Similarly, sodium is less in intracellular fluid and it is more in the extracellular fluid. And basically, this charge disequilibria is responsible for the generation of the resting potential at resting state. Now, so let us discuss the role of various ions in generating the resting potential. So first of all, we'll discuss about the role of potassium in generation of resting potential. Cell membrane is a semi-permeable membrane. It permits the passage of some ions and resists the others. Although the size of sodium ion is smaller than the potassium, the hydrated potassium ion is smaller than the hydrated sodium ions. In aqueous solution, water molecules get attached to the ions and the ions get hydrated. Or in the case, if we compare between sodium and potassium ions, we find although the sodium ion is smaller, but in the case of if we compare sodium and potassium hydrated ion, the hydrated potassium ion is smaller than the hydrated sodium ion. So potassium can move more freely between the two chambers, between inside of the cell and the extracellular fluid. Hence, the permeability of the potassium ion will be more in comparison to that of sodium ions. Potassium ions move from intracellular fluid to the extracellular fluid and vice versa. But because of the high concentration of the cell, potassium ion concentration inside the cell, there is net efflux of potassium ions. The motive force being the higher osmotic pressure of potassium inside the cell. Now, the shifting of cations across the cell membrane results in the membrane capacitor becoming charged and gives rise to membrane potential. The membrane potential acts in such a way that it opposes the efflux of potassium taking place due to higher osmotic pressure of potassium inside the cell. So, a stage comes when the influx and efflux of potassium are balanced, and it is referred to as the equilibrium potential of potassium, or EK. So, what is EK? Let us discuss what is EK. The quantitative relationship between the concentration ratio and equilibrium potential is called EK, or is called Nernst equation, or Nernst equation represents that, the quantitative relationship between the concentration ratio and equilibrium potential. The potassium equilibrium potential is determined by the concentration ratio of potassium ions inside and outside the cells. So, if we try to determine the equilibrium potential of potassium, it comes to about, or EK. If EK is the equilibrium potential of potassium, its value will be minus 61 millivolt into log Ki by Ko. Ki is potassium concentration inside the cell, and Ko represents the potassium ion concentration outside the cell, and minus 61 millivolt is a constant derived from the factors. Minus 61 millivolt is determined by the several constants, that is RT by Zf. R is the gas constant, T absolute temperature, Z is the valency of the ions, and F is the Faraday's constant. So if we put the value of Ki by Ko, which is 30, suppose concentration of potassium ion is 30 times more in the intracellular fluid in comparison to that of extracellular fluid, and we put the value, it comes to around minus 90 millivolt. And our resting potential is minus 75 millivolt. So, it is the resting potential of neuron is nearer to the equilibrium potential. So, sodium and so here we are studying the role of sodium in generating the resting potential. The dependence of the resting potential on potassium can be checked experimentally. The extracellular Ko value can be varied. How can we vary it? Because if we carry out in vitro experiment, we can always change the concentration of Ko or Ko outside the cell. Now, in addition to Ko values, Ek can be calculated with Nernst equation. So, if we, for different Ko values, Ek can be calculated with the Nernst equation, and it will come as the straight line. But at the same time, we can even measure the membrane potential by inserting the electrode. And we see that the graph below shows that only in the upper region of the curve, that is at higher Ko values, up to this Ko values, do the measurement agree with the theoretical relationship, that is Ek. While at low Ko values, they deviate upwards. So, at the low extracellular Ko concentration, which is actually in the case of nerve cell, the resting potential is less negative. This is also true for the normal extracellular concentration of 4 millimole per liter, is the normal extracellular concentration of the potassium in natural cases. So, at this value also, the membrane potential doesn't tally the calculated Ek value. It is slightly less negative than the calculated value, and that means some other ion is taking part in this region of the graph, or at this Ko value. So, if the same experiment is carried out in a bathing solution where sodium is replaced from the extracellular fluid, if we remove sodium, and we replace sodium, that the positive sodium ions, with some other positive choline. Choline is a protein positively charged, and why protein is taken? So that it cannot cross the cell membrane, or a big molecule which cannot pass through the membrane. We find that resting potential tallies exactly as the value of the EK value. So if we remove sodium ion, we will see that the resting potential will be same as the calculated, the membrane potential will be same as the calculated resting potential. So this means that the ion which is taking part in this deviation is sodium ions. And this observation shows that sodium ion is also influencing the value of the resting potential. So, if NaI plus NaO, if the ratio of NaI means sodium ion inside the cell and NaO sodium ion outside the cell is 1:12, then according to Nernst equation, ENA will be minus 61 millivolt log 1 by 12, or ENA is equal to plus 65 millivolt. And I have already told you earlier that minus 61 millivolt comes from derived from various constant, ZF, RT by ZF. So the value of ENA is plus 65 millivolts, and we know the value of potassium EK was plus 90. The role of the sodium ion taking part in generating the resting potential. So now it is clear that the ions which are responsible for generating the resting potential are mainly potassium ions and also to some extent the sodium ions. So these two ions are just significantly, and several other ions are also taking part, but these two ions are significantly responsible for the generation of the resting potential. Now, let us come to sodium potassium pump. What is sodium potassium pump? So, till now we have come to know that it is the tendency of potassium ions, as the potassium concentration of potassium ion is more in the intracellular fluid. It is the tendency of potassium ions to move to the extracellular fluid. And similarly, it is the tendency of sodium ions to move to the intracellular fluid. So this might cause the charge disequilibrium. So the excess of potassium ion which has moved out and excess of sodium ions which has moved in are brought to normal compartments with the help of sodium potassium pump. So the potassium ions have tendency to move to the extracellular fluid, and sodium have tendency to move to the inside of the cell. Now the excess of potassium ions which have moved out and sodium which has moved in are brought back to their original compartments with the help of sodium potassium pump. As the movement of the ion is against the concentration gradient, there is always involvement of energy. In biological system, energy is used in the form of chemical energy. It is liberated through the breakdown of ATP into ADP. Pi is inorganic phosphate, and this energy of the reaction is released, which is consumed in the reaction. Or ATP is equal to ADP plus Pi plus E. E is the energy. X and Y. This is the diagrammatic presentation of sodium potassium pump. X and Y are enzyme carriers present on the membrane. X and Y are the membrane. They are protein in nature, and this is the diagrammatic presentation of the cell membrane. This is the inside of the cell. This is the insulating membrane having all the carriers X and Y, and this is again exterior of the cell. So from inside. Inside has become there is excess of sodium ions. So sodium gets attached to the Y complex. Y is a carrier protein. It combines with sodium to form NaY complex, which diffuses to the exterior side of the membrane. And here NaY dissociates. Na is liberated into the extracellular fluid and Y is free. And now with the help of the enzyme, Y is converted into X. And X has again capability to combine with the potassium ions. Potassium ions which has actually moved to the extracellular fluid. And these K potassium ion combine with X to form KX complex, which diffuses to the inner side of the membrane. And here the KX dissociates into X and K plus. K plus move to the inside of the cell and X is again released. Now, so as to convert X into Y so that it can bind with another sodium molecule, there is involvement of energy, and uh, actually in conversion of one X into one Y, 1/3 ATP is required and it is converted into 1/3 ADP, and energy is required by the reaction. Why it is 1/3? Because one ATP molecule is sufficient for the conversion of 3X molecules into 3Y molecules. So this is the role of sodium. So these three things are taking place, or three phenomena are taking place for the generation of the resting potential. The role of potassium ions we have discussed, role of sodium ions, and then again sodium potassium pump. So this was about the resting.